Hi folks, Lisa Kirk here. I'm the principal biogeochemist with Environment. We're a division of Respec, located in Bozeman, Montana, about 6 hours south of the Elk Valley, where this work with Tech Resources and now the Elk Valley Resources Group, studying in situ biological reduction of selenium and nitrate in coal mine saturated rock fills has been undertaken by myself and my co-authors. I'm sorry I wasn't able to get to Berlin to be with you in person due to a family emergency. I do want to share with you, however, what we've learned because we're excited about this robust and resilient approach to mine water management. In this case, we have green injection wells and red pumping wells. The red pumping wells pull water away from the injection wells in both directions. And then the yellow and the orange are wells that were constructed for monitoring purposes. We monitor in these locations both chemistry using pumped groundwater, but also with biocoupons that let us measure the microbial community. This facility has been in operation for 8 years, originally as a field scale trial facility and now as a commercial facility, which treats 20,000 cubic meters of water daily to reduce 99% of the nitrogen and 97% of the selenium, and in doing so, reduce the client's cost of water treatment by about 80% on CapEx and 50% on OpEx. The facility has a 30-day residence time, roughly. The treatment zone is approximately half a kilometer long, and the thickness of the saturated zone in the subsurface is about 40 meters. Treatment happens within a relatively short period of time. We'll have a look at our information about saturated rock fills from column and field scale trials. Here we see nitrate and selenium affected water being introduced via pumping with nutrient into the saturated fill, which has an oxygen gradient. Nitrate is ideally being converted to nitrite and nitrogen gas, which is inert. Selenate is being converted to selenite and elemental selenium, which is insoluble. We are using the native bacteria to accomplish this, and we've done a variety of studies at the column and bench and field scale to understand operational performance, upset responses, response to closure, reversibility, and longevity of biomineralization, speciation of these compounds, etc. So let's have a look. Selenium occurs in mine water as both selenate or selenite, though these are both the more oxidized and more mobile forms of selenium. Once selenium is reduced to zero valent selenium or to selenide, it becomes immobile. Our goal is to reduce selenium to an insoluble form, such as elemental selenium, but absorption of selenite is also helpful. And we want to do this with the native communities. We want to create long-term stable sequestered selenium products. Here we have rock that has been weathered in a column in our laboratory over a period of 3 years. We can see the red elemental selenium that's formed on these mineral surfaces with the white biofilm associated with the growth of the bacterial community that supported the reduction of this selenium. We collected samples through sonic drilling and built replicate columns. We used site water and amended it with methanol and phosphoric acid. In an upflow configuration, we saturated these columns and we collected effluent for analysis of multiple parameters on a weekly basis. We obviously looked at DOC and phosphorus, but then again, nitrogen, sulfur, selenium, other dissolved metals, some of the geochemical parameters, redox, oxygen, pH, and conductivity. These tests are conducted in a cold room so that we can replicate subsurface conditions around 10 degrees C. And here are some of the pumps that we use for feeding that water. Results show that it takes a while for that biofilm to develop, and we see the first 100 days or so that the carbon's being significantly consumed by that effort. And we see that we have significantly noisy conditions with some selenium reduction. We're adding 1 milligram per liter, but in this case we've got still probably only 40 or 50% removal, maybe 60. And so we see that we don't really settle down in terms of selenium removal until we get out to about 150 days. So it takes a little while for things to develop and settle down. We can see that that was true for nitrogen as well, although it achieved steady state and strong removal sooner. We did have an upset condition in the column tests at week 320, and we can see that it took the columns a little while to reestablish their equilibrium, but they did. And the same is true for both nitrate and selenium. We can see that carbon use increased during that upset period. So we relied less on the carbon when things were at steady state and running nice and smoothly once the biofilm had been established. Once we were done with these columns, we decommissioned them and we sampled at the influent base, the middle, and the top of the columns. And we used 16S genomic analysis of the 16S ribosomal RNA gene to compare the relative abundance of microbial communities. And here we are showing things organized by phylum, which is pretty high level, but basically we're showing the blue aerobic heterotrophs that are consuming carbon decreasing as we move away from the point of carbon injection, which makes sense. Likewise, the organisms that specifically are using methanol, like Methylotenera, and some of these others that fall into the proteobacteria are declining somewhat as we move upward in the column, and we are increasing in abundance of some of these sulfur-reducing bacteria, the purple, and desulfurosporinus. So we see a shift in community as a result of the availability of nutrient. That's what we would expect. If we look at the full-scale SRF, we see changes in the concentration in the field scale trial over time. So here we have days and during startup, and here we have the selenium concentration in micrograms per liter and nitrate in milligrams per liter. This is a log scale. So the black is the influent concentration, and then these are averages from those colored monitoring arrays that we showed you in the aerial photograph. So we see that after about 100, 120 days, we began to see significant selenium reduction. We did have that upset condition, at around 140 days, and we see a temporary slowdown in selenium removal, but then it picks back up, same with the nitrate. So we can see that the system's resilient, that it can be upset, but once it's upset, it can also resume operations. We see, as we did in the columns, that the carbon was used to build the biomass and again to reestablish equilibrium during the upset condition. In both cases, we used bromide to as a tracer so that we could understand that we were getting all of the water back. And as you can see, we did pretty well. If we look at this normalized to 1, which would mean we got everything back, you can see that most of the bromide was recovered. Having the tracer allows us to calculate concentrations at C over C naught, so the effluent chemistry over the influent chemistry as a ratio. And what we can see even more clearly is that selenium reduction, the nitrate reduction, and the influence of those upset conditions in those later weeks. We were able to drill a hole, sonic hole, into the treatment zone. It took us a bit to get actually into that zone because the treatment was so effective, it happened so quickly that we had to really get in close to our injection wells, and initially we didn't want to disturb them, so we stayed farther out, and we found that we weren't intercepting the active treatment zone. And by that I mean that that we actually had no nitrate and no selenium left in solution. Makes it hard to calculate a rate. Here we can see that the water table's at about 57 meters below ground surface. This is a core, a drill hole that we constructed and completed as a monitoring well, but took the sonic core and sampled it. So we can see in blue where we have increasing mass of DNA per gram of sediment, and in yellow or orange where we were not able to recover any DNA. Above this zone at 61 meters, we have a vadose zone, or a zone where we have variable unsaturated and saturated conditions. So we don't have much biomass there, but immediately into the saturated zone, and where the majority of the methanol would actually float, would rise within the system, we do see the highest values of nanograms of DNA per gram of sediment, as much as 15,000 grams. And so that's quite a yield, and you can see that then it begins to diminish as we increase with depth and we leave the treatment zone entirely and get back into a zone where there was really no DNA yield. The highest microbial biomass coincided with the predicted active treatment zone based on modeling in that 62 to 67 meters below ground surface range. When we look at this from a diversity standpoint, we can see that we had higher diversity in the vadose zone where biogeochemical conditions are fluctuating, and below it where things are in their native state. But in the treatment zone itself, because we're applying a selection pressure, because we're adding nutrient, we're actually selecting for certain organisms, and the diversity decreased in that zone. When we compare the difference between microbial community structure in each sample, each of these is a sample depth, and the distance between points on this NMDS plot is showing you the magnitude of distance between or variation between microbial communities in each sample. So here we see that the active treatment zone samples are all clustering together in one area, and quite different from the zone beneath the treatment zone and above in the vadose zone. So using this dissimilarity, Bray-Curtis dissimilarity index with these taxa data, we were able to really distinguish the three zones with respect to the treatment area. And when we looked more closely at a sort of functional level, we lumped the samples or the identified microbes together based on whether they were methanol oxidizing or nitrate reducing or selenium reducing, so blue and green and red, or sulfate reducing. And then we plotted those results as a function of depth. And two things jump out. One is that we saw an initial increase right at the water table of the denitrifying and methanol oxidizing organisms, suggesting that methanol oxidation is supporting that nitrate reduction. Shortly thereafter, with depth, as water moves down, we can see that we are getting the highest abundance of selenium reducers right at about 65 meters below ground surface. Interestingly, we commonly see that selenium and nitrate reduction overlap, but that selenium lags just a bit behind that nitrate. We see that sulfate reduction was most active in the zones immediately above and below the treatment zone, and that's because the oxygen gradients there are the steepest, and we know that sulfate reducing bacteria really can't tolerate a lot of oxygen, so they're finding niches in these zones where the gradient's steep to accomplish sulfate reduction. We did see other organisms, iron cycling, hydrocarbon degradation, but we don't have time to get into that today. Instead of thinking about it, though, from a depth perspective, we can also think about it from an injection point perspective. And so here what we've done is plotted the similar analysis 16S genomics from biocoupons, which are little steel cages that we hung in the flow field in those monitoring wells. We did this, and we changed them every 3 months over a period of 5 years. We have quite a nice data set. Here at the 0 point is the point of injection. We're showing the relative abundance of organisms, and we've lumped them again. So we have the aerobic heterotrophs in blue, which spiked initially, used up that oxygen, and then became less abundant. Thereafter, we begin to see the denitrification kick in, and we see that it continues. And that makes sense too, because the mass of nitrogen is so much greater than the selenium, which kicks in a bit later, as we noted on the depth profile, but begins to tail off within about 30 meters. So we're accomplishing this treatment within a short distance of the point of injection. We do track iron and sulfur because those are indicators that we may over-reduce the system. We don't need to over-reduce the system. We simply need to have a suboxic condition. And so we can also here see the influence of those steep oxygen gradients at the point of injection and just below the treatment zone. So here we're laterally moving through the treatment system, but it looks a lot like that depth profile. We did use metagenomics, which allow us to characterize more than the probable organisms that are present and to infer their function. Rather, this lets us extract all of the DNA from a sample and then to look at the specific genes that are present and understand using a shotgun metagenomic approach, assembly of the reads that we get into contigs, and we bin those, and we can actually build some genomes, which are hidden beneath my little picture here. So better technology for understanding the genomic capacity of these samples to do the work we're asking them to do. Same figure again with that drill hole, depth from 55 meters below ground surface to 85. What I'm showing you here is that we chose 3 samples in red, the red bars, from 62, 65, and 70 meters depth for this metagenomic analysis. So this is a lot of data. It takes a lot of time. You can see that we got lots, 5 to 10 million reads from these samples. We assembled somewhere between 53 and 62,000 contigs, and we were able to get an awful lot of information at the gene level, which genes are most abundant, and indicating what processes are possible and perhaps happening. And then it also allowed us to assemble some of the genomes themselves. It also let us map the pathways that are important to the denitrification and selenium reduction. We were more successful with this with the nitrate, and that's really because more is known about denitrification. There's more information in the literature about the enzymes involved. So what we've done is shown here nitrate being reduced to nitrite, nitric oxide, nitrous oxide, all the way to dinitrogen gas. And then the bubble size indicates the relative abundance of key genes that we know are involved in nitrate conversion to nitrite. That's these NAR genes. Nitrite conversion to nitric oxide, that's these NER genes, and so forth. And then you see the incomplete reduction leading to some ammonium accumulation, which is, we see a little bit of capacity for that in the system as well. So this is really great because it lets us map the system and it lets us understand which genes are present and maybe being put to work. If we were to have an upset where we had a real problem with function, we'd be able to look back at these genes and their expression and understand what we've lost and what we need to fix. So this is a really useful tool. We're less able to do this with selenate reduction because there's so little known about selenium reduction. The selenate reductase enzymes that we do know about come from an organism called Taura selenatus. There's 3 different genes, SER A, B, and C. They're very similar to nitrate reductase. Enzymes known as NAR, G, H, and I. And so we don't have as many of the known genes to use in our mapping, but we were able to use the patterns that underlie these the sequence patterns from these genes, and build a database that lets us understand a little bit more about what may be happening in our system. And so What we've done with these same 3 samples that we've talked about is looked for a sequence that is similar to the SER-A gene, the SER-B gene, and the SER-C. And then these are the different elevations, 62 meters, 65, and 70. So you can see we have quite a few organisms that appear to have similar capacity genes in organisms that are present that seem to have capacity to accomplish this reduction. I don't really have time to tell you about our closure study today, but I do want to tell you that we've put quite a lot of effort into that because, of course, once we stabilize the selenium, we want it to remain stable so that it's not an environmental problem going forward. We've conducted a column study of the SRF biomineralization that accreted in the columns when we ran them over a period of months and years. We simulated closure by turning off the water and turning off the food, the carbon supply and the phosphoric acid. And then we watched to see if we would observe any release of nitrate or nitrite or selenium. Now, I'll just show you how we did this. Every 6 months, we would push 1 syringe, roughly 180 mils of water into the column. And because it was such a small volume, we weren't mixing through the column. We were just displacing the next 200 mil volume out the top, in essence. And so it took us a few years to push most of that water through. You can see in the shaded box, that's kind of the limit of that period of time when we were displacing that initial pore volume. And what we can see is there were some upset at the time, not terribly significant, but some, at the time that we stopped the flow of nutrient, and then it settled back down. The other thing that's really interesting here is that the site water has nitrate and selenium in it. And so we can see that even though we're not feeding these columns, there's sufficient carbon and biomass being recycled so that the system is able to continue reducing nitrate and also selenium. So we don't see reversal, we don't see remobilization, and we do see ongoing capacity to handle small amounts. So let's say there was a storm and it rinsed some selenium into that closed SRF, which is no longer fed. These data suggest that this will continue to serve as a robust and resilient sink for those elements. This is the graphical abstract from our paper we published in Stoughton this year about the closure work. I guess I'll just point to the mineralogy work that we've done. We have shown that the mineral precipitation and sorption that occurs, we see a precipitation of selenosulfide complexes, and we see sorption of selenite to the iron oxyhydroxide minerals. We know that selenate won't sorb, but selenite will in this pH range. And so we showed not only did these removal mechanisms occur, but they also were not reversible. We tried to reverse them under changing oxygen and nitrate exposure, pH, and we were unsuccessful. So we're quite confident that once this is reduced and made stable, that these materials will remain stable post-closure. In conclusion, our successful selenate and nitrate reduction in column and field scale saturated rock trials has shown us that this is possible, that it's cost effective, that it's robust and resilient and sustainable. We have shown that selenium is accreted in 2 forms, sorbed and precipitated. Both forms are stable under post-closure conditions, and this technology has now become commercial with a technology readiness level of 7 under review for level 8 in British Columbia. These are some publications which we would suggest you turn to with further questions, but I am happy to speak with any of you offline, by phone or online, however would be convenient. I'm sorry I wasn't able to be there with you. Thank you.